Hi, I'm Cal Parks.
And I'm Kim Gutjahr, and this is Cell Action News.
Now today, mitosis has just occurred in our cell.
Yup, mitosis is very important in cells, and that's why we're so excited about it. It helps the organism with growth and repair. And for unicellular organisms, mitosis reproduces the entire organism. Mitosis also enables multicellular eukaryotes to develop from a single cell.
Now let me tell you folks about prophase. This is when the chromatin begins to condense by coiling into discrete chromosomes, and the nucleoli disappears, and the nuclear membrane breaks down. The centrioles migrate to opposite poles, and the chromosomes are moving to the center of the cell. This is also when the mitotic spindle begins to appear. Isn't that exciting?
Yes, it is, but not as exciting as metaphase.
Well, what's that?
Metaphase is when chromosomes take up positions on the metaphase plate. Each double-stranded chromosome is attached to a spindle microtubule at its centromere.
Yep, that's right, the shortest stage of mitosis. Anaphase is when the centromeres of each chromosome separate each sister chromatid and become a chromosome, and now there are twice as many of them. The spindle microtubules attach the centromeres of chromosomes to move the chromosome, and when a chromosome without centromere is just left behind in one of the two cells. This is also when our cell elongates and the chromosomes are each in both sides of the cell.
Telophase is when chromosomes gather at the poles of the cell and form identical daughter nuclei, and the nuclear membrane begins to reform from fragments of the parent's nuclear envelope and from the endomembrane system. Chromosomes become less condensed, and the nuclei resembles interphase again. The division of the nuclei is now complete.
And now for the piece of resistance, folks, cytokinesis, which is just a fancy way of saying the division of the cytoplasm. And this is when the cell, like an animal cell for us, would pinch off in the middle and the two daughter cells would become separate entities. But in plants, plates begin to form and cellulose is laid down on each side, creating cell walls, creating the division between two new daughter cells. And they each have the same chromosome as the parent. Isn't that weird, huh?
Yeah, they're identical.
All right, now we're going to go up to our action reporter, Jim. Well, hi, Cal. Thanks for checking in. We're going through mitosis here as well. But there are some weird things happening. In fact, prophase, which had just occurred, chromosomes have condensed just like normal mitosis, except they've been crossing over and swapping genetic things between non-sister chromatids over corresponding segments of DNA. I don't even know what that means, but I was just seeing it happen right here, and we've gone right on into metaphase, exactly the same, but anaphase. There's a breakdown of proteins keeping the sister chromatids bound, allow the homologs to separate. Homologs move towards opposite poles, guided by the spindle apparatus. And generally, that's all the same down on the telophase, and that's all happening the same too. The two cells are pinched off, and we're just completely confused right now because things start to change around here. And people don't even know how to spell mitosis. You see that? What's happening? What's going on? Jim, we're getting a live report here. In fact, what Jim has just gone through is meiosis, which is a separation of homologous chromosomes through the first phase. Where the prophase that he was experiencing was indeed a condensation of chromosomes into loose pairs of homologs that connect through synapses and the crossing over of genetic material. However, he also seemed to miss that there was a chiasmata which existed at a crossover of the chromatids. And the metaphase was exactly the same.
Okay, and in anaphase, it is the breakdown of proteins that keep the sister chromatids bound, allowing them to separate. However, sister chromatids' cohesion is still at the centromere, which allows them to move as one to the same pole. Telophase is also the same as mitosis, and cytokinesis is also just the pinching off of the two cells.
And then we move right on into meiosis 2, where the sister chromatids separate. And prophase 2, exactly the same. Spindle apparatus forms and moves toward the metaphase plate, where the chromosomes take position, and but except the sister chromatids are not genetically identical. Kim?
And then in anaphase, there's a breakdown of the proteins that bind the sister chromatids at the centromere, and the chromatids separate, moving toward opposite poles. Then during telophase 2, the nuclei form, and chromosomes decondense, and cytokinesis occurs.
From the results from this meiosis is that 4 daughter cells are formed that are all genetically distinct from each other and the parent cell, and have half the amount of chromosomes that their parent had.
Okay, so both meiosis and mitosis involve chromosome replication and the cells splitting apart, and they both create new daughter cells. Meiosis occurs in reproductive cells and consists of 2 nuclear divisions. It involves the separation of homologous chromosomes in M1. The diploid number of chromosomes is reduced to haploid in each daughter cell. Chromosomal characters are altered due to crossing over, and that causes changes in genes. And in mitosis, it occurs in somatic cells, And there's only one nuclear division, and it involves the division of chromosomes. There is no pairing of homologous chromosomes or crossing over, and the number of chromosomes present in the mother cell is maintained.
Okay, folks, you stay tuned. Till then, Selection News.